Hi, my name's John Fuller, and I'm the application scientist for Labcyte, covering the New England area based in Boston. And today I'm going to talk about applications in synthetic biology, but frankly, given the previous session from the Foundries, I don't know that I can do a better job at highlighting our applications, but I can't let you go to lunch just yet. So I figured I'd talk a little bit about what goes on under the hood, so to speak, with echo technology. For those of you who are unfamiliar with the echo, what our instrument does is use acoustic energy, which converges at the air-liquid interface to form a mound of fluid. And from that mound of fluid, depending on the instrument, we are able to generate 2.5 or 25 nanoliter droplets with high precision and accuracy. And so with our machine, you can move from any source containing your sample to any destination without ever touching the fluid. Our proprietary technology, dynamic fluid analysis, is the key to successful aqueous transfers. This graphic illustrates how this works. The echo sends a small ping of energy into the well. That energy reflects back, and then from that reading, we are able to generate and to understand the acoustic properties of that fluid. And this enables us to dispense any volume within range given a wide variety of fluid properties. And our calibrations are turnkey ready and fairly straightforward to use. So if you're familiar with the history of the echo, we got our start on high throughput screening platforms, dispensing small molecule libraries. But with the advent of dynamic fluid analysis, we've been able to push the envelope and leverage the technology in molecular and synthetic biology. Probably the most widely used molecular biology application for the echo is tipless, touchless, PCR, and qPCR. The echo can be used to dispense master mix, cDNA, primers, and water backfills in a simple single protocol. And you can do this with either our application, which is a fairly straightforward graphical interface, or you can also code this and use a CSV pick list. This heat map from 1536 well light cycler highlights the abilities of the echo to target wells without touching them and without any cross-contamination. So what we've done here is fill the entire plate with master mix and primers, so we're ready to go, but then we've only dispensed the DNA to the wells highlighted in green, so you see no cross-contamination in the wells highlighted in red. Moreover, we are able to dispense 500 nanoliter qPCR reactions with sub 2% CVs on the CQs. The next few slides are highlights from the echo's use in the 2016 Cold Spring SynBio course held last year. Instructors Mike Smansky and Vincent Neuro used the echo in their respective modules. And they've been kind enough to provide me data from those modules. And just as a shameless plug, the Echo 550 will be on site at the 2017 Cold Spring course. So if you know of a student that's attending the course this year, please send along a very complicated pick list for us to try. Our typical runs are between 15 and 30 minutes, so I get bored very easily and would like a challenge. So in Mike's module, the students compared Gibson and Golden Gate assembly using the echo, and were able to get colonies from 2, 3, or 5 fragments with high efficiency. And they were also able to miniaturize the reactions down to about a 250 nanoliter reaction with high efficiency at 5 fragments, putting them together. This is fairly routine if you are a DNA foundry, but these were students with little to no assembly experience. So they were able to knock this out, and using something like Golden Gate, they were able to permute the elements at will, given our ability to dispense from any well to any well. In Vincent's module, Vincent highlighted the use of the TXTL reaction, transcription translation, which is a great breadboard for being able to test gene circuits in a cell-free manner. What we were able to do with his lab, as well as the in the course, was dispense all of the components necessary for the TXTL to take place. And given this example data that he generously provided, we were able to change the stoichiometry of the gene circuits and then read in real time the production of the GFP. Subsequently, Vincent purchased his own Echo, and as he noted in his lab impact, they've been able to increase the throughput, but more importantly, they've been able to gain significantly in the reproducibility. Every Echo run, you get a transfer report that tells you what you did. So you've created your construct, you've maybe tested it in a cell-free system. We've also, our customers are using the Echo in NGS, particularly in library preparation, quantitating for NGS, and pooling and normalization. One example from Amyris was the ability to microscale the Nextera reaction from 50 microliters down to 500 nanoliters, adding 200 nanoliters of DNA with the Echo, and then the tagmentation master mix, 300 nanoliters, and then being able to terminate that in also with the Echo, and then also using the Echo for adding the primers. And so what this has enabled is nearly a 100-fold reduction in the costs of the tagmentation reaction, from $73 per reaction down to 73 cents. And with our ability to dispense tip-free, we're also able to reduce the costs and the volumes significantly in reactions such as PCR, as well as Gibson Golden Gate. So I'd like to leave you with the key benefits and features of the Echo. Most notably, we're able to dispense at 2.5 or 25 nanoliters. This enables us to save reagents and miniaturize your assays. Our technology is non-contact, and it's a very gentle technology. We can actually dispense cells with it. We can do any well to any well, so there are no fixed tip constraints. And most importantly, even though the Echo is used on expensive automated high throughput platforms, DNA foundries, I think it's fairly end user friendly, and I think I can teach the average person to set up an Echo transfer and drag and drop their liquids around in about 30 to 40 minutes. Of course, I've only been able to scratch the surface of our applications in 7 minutes, so I would invite you to visit labcyte.com and our resources page, and I'm happy to take any questions. Thank you for your time.